EU-OPENSCREEN is a multinational European infrastructure of open screening platforms and chemistry sites for chemical biology and early drug discovery research. You can find more details on EU-OPENSCREEN and its operations through our website, which will be mentioned at the end of the webinar. Thank you all for taking time out and being here today. I'm Kathy Skopelitou, the training manager here in EU-OPENSCREEN, and in today's webinar, we'll be looking at the following. Our first talk from Professors Mabel Losa and Ángel Carajedo will introduce us to today's topic, emphasizing at the challenges of DNA encoded libraries. Dr. Eduardo Dominguez will then give a speech on the fundamentals of library production and affinity selection, as well as the description of the workflow. The next topic will focus on how hits can be identified by DNA sequencing, presented by Dr. Beatriz Sobrino, and our final speaker, Dr. Jorge Amigo, will give a very interesting talk on data analysis and more specifically on data normalization and detection of binders. Application exams will follow right after our last talk. I would remind you that this webinar is being recorded and therefore all attendees are muted and the camera pictures are hidden. However, you will have a chance to submit any upcoming questions through the questions and answers icon window throughout the webinar. There is also the possibility to ask questions anonymously for you to feel more comfortably. You can find this Q&A icon on the bottom of your screen in the Zoom window, so if you have any questions, just pop them in there. At the end of the webinar, we will go through the collected questions and the speakers will answer them. And if you miss anything, don't worry, we'll be sending around a link to the recorded video when it is available. At this point, I will ask our first speakers to start with their talk, but before we do this, a few words about them. Mabel Losa is a professor of pharmacology department and head of biopharma pharmacology research group at the University of Santiago de Compostela in Spain. Leader in the field of early drug discovery at Spanish and European levels, having active participation in the development of 15 new chemical entities that have reached clinical trials in the specific programs of research and development transferred in drug development in different pharmaceutical companies. She is the coordinator of the Drug Discovery Galicia Network since 2007 and the Drug Discovery Spanish Network of Excellence, as well as the Spanish OpenScreen network connected to the ERIC EU-OPENSCREEN. Our next speaker, Ángel Carajedo, is a professor of legal medicine at the University of Santiago de Compostela in Spain. He is also the director of the Galician Public Foundation of Genomic Medicine and director of the Spanish National Genotyping Center. He has been a committee member in numerous international societies for genetics, pharmacogenomics, cancer, and forensic medicine, as well as a member of numerous advisory committees of research centers and regulatory bodies. He has published more than 600 articles and is the most cited author in the field of forensic medicine. He has received many prizes and has been nominated Doctor Honoris Causa by various universities in Europe and Latin America. Mabel and Ángel, we are looking forward to your talk and sharing with us your expertise on today's topic.
Thank you for this nice introduction. Good morning and welcome everyone. Thank you very much for participating in this webinar today. It allows us to share with you our experience with DNA-encoded libraries for undruggable targets, which is very complementary to the classic chemical biology and drug discovery, to have chemical proofs and/or first hits in drug discovery. Therefore, it has a space and an utility alternative to a classic hit search campaign in difficult targets. It's a way to find hits in those mechanisms of interest that do not respond in attempts to classical screening methods. It's based on joining big numbers of molecules at each point of checking in the target. In some ways, it is the approach that is used for combinatorial chemistry to increase the numbers, to have more probabilities to the additional to the study of SAR or the rational based studies. Regarding this comparison with combinatorial chemistry, in terms of associating thousands of hundreds or thousands or million of compounds at the same time against a target, with the advantage over medicinal chemistry that allows the active ingredients to be separated very easily. This is the trick. What does the use for this separation? DNA tags. DNA labels allow you to mark millions or perhaps hundreds of millions compounds. This label is the sequence of DNA and is based on DNA in our and every DNA sequence of life. The separation of the assets then is done very easily with genomic sequencing methodologies. So this method is something like fishing to find active molecules against undruggable targets, the possibility of testing millions of molecules in a single assay, and separating the active molecules in a very simple way through sequencing. This is very cheap and very fast, but this is a large number approach. Reproducibility is not usually so like in our classical screening campaign standards, but rather to verify that hits are really hits is necessary to come back to the screening methodology we call affinity selection. So for us, our collaboration with the genomic expertise of Ángel Carracedo team allow us to complement our pharmacology knowledge in this coordinated Innopharma platform at the University of Santiago de Compostela. And we learn with Ángel Carracedo team's DNA encoded libraries together by doing it. We had the great opportunity to doing that because the Eli Lilly companies commissioned us with a first job years ago. So we acquired experience and knowledge with Lilly researchers, and it was a great opportunity that is very complementary to our EU-OPENSCREEN library to mark disruptive targets with that library. We are, of course, very happy to share our knowledge and to answer any question from you now and whenever you want. So just let me to introduce Ángel Carracedo, the co-coordinator with me of our site here in Santiago de Compostela. He directs the Galician Foundation for Genomic Medicine, as it was mentioned in the CV, and he is one of the best geneticists in Europe, a wise man and a professional and a person from whom I learn every day. I pass the intro to Ángel Carracedo.
Well, good morning, everyone, and thank you so much, Mabel. You are always so kind. And very briefly, more than 10 years ago, we started a joint venture of two groups, the Mabel group and our group on genomic medicine, with the idea of creating a pharmacogenomic platform and accelerating the process of drug discovery. Until very recently, the contribution of the geneticist, that's to say our group to this pharmacogenomic platform, was mainly in the target discovery phase using genomic approaches, as well as in the identification of genomic biomarkers for drug response. That's to say pharmacogenetics and pharmacogenomics. But the introduction of the DNA encoded chemical libraries offers us a new possibility of collaboration. It's an amazing technology from my point of view that takes advantages of the properties of the DNA molecule and the advances of the DNA sequencing technologies. And it's so nice to see that now we have new possibilities of collaboration, of interaction between our two groups. Indeed, DNA technologies are accelerating the drug discovery process, and in particular the early phase discovery activities such as target validation and hit identification. So I think you are going to enjoy a lot this webinar. Although Katia has already presented the speakers, let me to introduce them again. Eduardo Dominguez, Dr. Eduardo Dominguez is pharmacologist, is senior research in our groups, is the real link between Mabel and me, and in our two groups. Beatriz Sobrino is the director of the sequencing platform of our group, and she has a great experience on next generation sequencing technologies. Dr. Jorge Amigo is the responsible of the bioinformatic platform of our group of genomic medicine, also with a great experience, and he has also developed tools for Dell. So I just want to thank the organizers, Katia, and all this European OpenScreen platform, and Mabel, as always, and the speakers, and of course, all the people that are attending the webinar. Thank you so much.
Thank you, Angel. Thank you, Mabel, for your interesting introduction and talk. We now have a precise and clear idea for today's topic and the challenges related to the DNA encoded libraries technology. For those of you just joining us now, welcome to today's webinar. I would remind all of you that we'll be running a live question and answer session at the end of the webinar. Through our question and answer icon window, which is on the bottom of your screen. So if you have any questions, just type them in there. As you said, Angel, our next speakers are Dr. Eduardo Dominguez from the University of Santiago de Compostela and from the Galician Public Foundation of Genomic Medicine. We are very pleased to welcome Dr. Beatriz Sobrino and Dr. Jorge Amigo. A few more words about our speakers in order to get to know them better. Dr. Eduardo Dominguez has a degree in pharmacy and a PhD in pharmacology. Since 2013, he has been the coordinator of the Biological Reagent Production Unit of the Biopharma Research Group, where he is involved in early drug discovery research projects. He has over 10 years of experience in developing assays and high-throughput screens by using a number of technologies, including DNA encoded libraries. Dr. Beatriz Sobrino has a degree in biology and a PhD in molecular medicine. Since 2009, she has been the coordinator of the DNA sequencing unit of the Galician Public Foundation for Genomic Medicine, where she performs tasks both in support of routine genomic diagnosis and research. She has over 15 years of experience in massive DNA sequencing data and over 5 years of experience in sequencing experiments performed with DNA encoded libraries. Our last speaker, Dr. Jorge Amigo, has a degree in physics, a master's degree in bioinformatics, and a PhD in molecular medicine. Since 2015, he has been the coordinator of the bioinformatics unit of the Galician Public Foundation for Genomic Medicine, where he performs tasks both in support of routine diagnosis and research. He has over 10 years of experience in analyzing massive DNA sequencing data and over 5 years of experience in analyzing DNA encoded libraries. So let's go directly to our next speaker's presentation. Eduardo, you may share your screen with us. We are really looking forward to your talk.
Well, thank you everybody for joining us today for this webinar with this interesting technology that we are going to share with everybody. and for the kind introduction as well. This is recapitulating some of the information that Mabel and Angel already introduced. Since we have experience in drug discovery and genetic analysis, we team up in order to provide this kind of combination of genetics and chemistry analysis in the pharmacology context. Since we have the opportunity to partner up also with the industry, we think that we have built a team that can tackle many of the technological challenges for the DNA encoded library technology, but also for the chemical biology and as well for drug discovery when these projects advance to a more mature stage. In that sense, I have to emphasize that genetic sequencing is used for variant identification, diagnosis of rare diseases, understanding disease, but also until once the next generation sequencing technology was introduced, was helping to decode the libraries that are synthesized in this way, combining a small molecule with a oligonucleotide that provides an identification, a unique tag for each of these molecules. And within this platform, we have been able to develop different technologies for designing and support the encoding methods, and also tools based on bioinformatics and algorithms that Jorge will talk later about, in order to perform better designs, more efficient analysis, quality controls, and sequencing workflows that ultimately have led to generate more than billions of reads per year and performing multiplexed analysis of different kind of libraries and for multiple targets. And here is shown the general workflow of this kind of screenings that we're gonna focusing on today, including the first step of designing and production of a library that I will mention briefly, but not going into very much detail, but rather providing an example. The affinity selection step, which is the step in which you display the moieties of the protein of interest, target of interest, against the moieties of small molecules in order to come up with binders or interactors. And then the encoding step, which is gonna be the DNA sequencing, in which Bea is an expert and will explain in more detail. And finally, the analytic part, in which Jorge will tell how to develop this kind of analytic workflow that will help to finally annotate the chemical structures in terms of biology, the biology of the target that you are studying or exploring. Of course, there are other steps further, but for today, we are going to be only talking about this, what we consider key steps on the process. Here is shown an example of a library which contains a chemical compound shown here in blue and coded as first building block, which also has linked a DNA tag that will identify this particular molecule once all the steps are performed. In this example, it's a double-strand DNA encoding method, which after this first step of chemical synthesis and ligation of oligonucleotides, one can go ahead again and re-synthesize a second building block or a second set of building blocks with a second DNA tag set, and start building what is considered a diverse and large pool of DNA encoded chemical compounds that eventually will be forming a pool of millions of compounds that can be, as mentioned, explore with potential targets. Finally, after these iterations of splitting and pooling the compounds, the dual compounds, we can assume that each of these small molecules has a unique DNA sequence and can be put in a test tube against a target. And in this case, we are showing here that if you immobilize a given protein in a support or by other means, such as solid phase or non-immobilized method, such as the available technologies you can test whether these molecules target the protein or bind the protein by incubating them in different conditions, including different concentrations of the protein, the tag itself that was used for isolation and immobilization of the protein in order to discard the artifacts of the non-specific binders, many different stringent conditions such as pH, temperatures, etc. And also compounds that provide you with a reference of a binding activity so that you can also establish the affinity of the new chemical matter that might be identified or bind this protein against the standards. And then you go ahead and select by doing or performing different rounds of washing the compounds that finally are not able to interact with the protein. And you have to select, of course, which is the optimal round in which you want to enrich your target with the library. And in order to get the codes of these molecules, you will use methods such as denaturing of the protein and isolate the binders or the DNA, which is finally what you get after the process of affinity selection. For example, precipitating the DNA with ethanol crash or other means, and submit the sample for further amplification steps with PCR, quality controls, and finally sequencing that will eventually provide you a sense of if within your library you have molecules that might target the protein, and you can further explore them by performing the next step, which are going to be to test them in different assays, either biochemical or biophysical or cell-based, that will provide you a reference of or a certainty that your compound is active. And here is showing an example of results coming from an affinity selection against a target protein. This is the final read counts obtained from the sequencer, and the size of each of these points represents the number of counts of DNA and is related to the affinity of these molecules against this particular protein. And on the left, you can see an example of what is molecule formed by the 3 cycles that was used for building the library, which means that it has the general diversity of the library, but if this singleton or this line is then resynthesized of DNA, you, after testing, you can confirm whether these these initial hits from the affinity selection have confirmed the activity. And in this case, more than 80% of them were positive. Of course, there are more optimization steps by medicinal chemistry in order to be able to optimize the potency, the selectivity, and the pharmacological properties of the compound. And on the right, you can see how a plane, an entire plane of building blocks is identified, containing not only the general diversity of the library, but an entire plane and several chemotypes that are unique of this plane. And again, after testing these compounds of DNA, more than 60% were positive, meaning that in this particular experiment, it was useful to apply this technology to explore the ligandability of the protein and generate new chemical matter with different diversity, which for a medicinal chemist is a very good starting point. And with that, I will stop sharing my screen and let Beatriz to go ahead with the DNA sequencing step and explain a little bit further. And later we can discuss some of the challenges that the remaining questions of the technology.
Hi everybody. Thank you for joining this webinar. Okay, so after affinity selection that Eduardo has already explained to you, we need to identify the compounds with affinity to the target of interest. And how we are going to do this? So, well, we are going to use the DNA part that is attached to the chemical compound to identify this compound, because each DNA part is specific to each DNA compound. So, well, the technology that allow us to do this are the next generation sequencing platforms that allow us to sequence from several to millions different DNA fragments at the same time. So, well, there are several DNA platforms in the market. We can see in this graph many of them. There are some news that are not represented here, and some of them that are represented have been already discontinued. But in general, we can have an idea that there are several platforms on the market. And well, in this graph, we can see two parameters that are important for the selection of the platform of interest for each application. So here we have the read length, and in the other scale, in the Y, we have the gigabase per run, the throughput. Okay, so we have massive parallel sequencing platforms with different capacity and also with different read length. Okay, so we need to have these parameters in mind when we select the ones that we are going to use for a certain application. We will see later which are the ones that we are using for DNA encoded libraries. Okay, so for DNA sequencing, in general, the workflow for all the applications is the first step is the DNA library preparation. The second step is sample pooling, in this case from different affinity selections, but in other cases from different patients, different experiments, whatever we are going to, we are interested in sequencing. And the third step is sequencing the next generation platform. Okay, so we are going to see how we prepare the library for DNA encoded library. So in this case, we are going to use a two-step PCR procedure. This method is going to use the strings of the DNA part of the DNA encoded libraries, because these strings are specific for each type of DNA encoded library. So they are all the compounds have this same sequence in these streams. Okay, so we are going to use these streams for PCR primer design. And because it's a two-step PCR method, we have to design two primer sets. The first primer set is the one that has a region that is complementary to the headpiece or to the closing primer. Okay, so in this case, we will use this part to design a complementary primer that has an overhang that is going to be complementary to the second primer, to the primers that are going to be used in the next PCR. Okay. So with these primers, we are going to amplify the products that comes from the affinity selection. The second PCR is needed to include to this oligonucleotide the primer, the sequence adapters of the next generation platform that is going to be used for sequence. Okay. So these primers has a specific part that is called index or barcode, depending on the platforms that we are going to use, and can be one or two, one on each primer. And this small oligonucleotide is going to allow us to sequence several samples at the same time. Okay? This is one of the advantages of the next generation sequencing, because we can sequence millions of fragments from the same sample, but at the same time we can also combine samples, different samples, because we are going to use this small region that is included in this case in the second PCR to demultiplex the sample in the analysis, in one of the analysis steps. Okay, so after the PCR 1 and 2, we will have a final DNA sequence that is going to be loaded on the sequencer. Okay, there are different PCR cleanups in between these steps, because we need to remove the rest of the primers from the previous PCR and the rest of reagents used in this step. But I mean, it's in some ways an easy DNA library preparation, easy and quite quick. Okay, so once we have the libraries prepared, we will have to do the sample pooling. And for sample pooling, we are going to use what I have mentioned before, that is the index and barcodes that allow us to sequence several samples simultaneously. Okay, so this means that we are going to have millions of reads at the end of the sequencing run, millions of reads all together. But with the bioinformatic analysis will allow us to demultiplexing them, and we can separate the reads that belong to one sample and the reads that belong to the second sample, to the first sample, etc. Okay, so this is very important because it's what it does. This kind of technology is so useful for all the applications. Finally, we will have the last step, that is sequencing the libraries, the DNA sequencing libraries in the NGS platform. As I mentioned before, there are several sequencing platforms on the market. In our case, we have experience with IonTorrent and with Illumina platforms. The first one is based on semiconductor sequencer, and the second one is based on sequencing by synthesis and fluorescence detections. Both of them have advantages and disadvantages, as everything in general. So, but in this case, the main problem of the IonTorrent platforms is basically the throughput. So the maximum million of reads that can be loaded on a IonTorrent platform is 130 million. So this is not enough for sequencing libraries with millions of compounds. So this is why Illumina platforms are the most used platforms for this application. So there are appearing new platforms in the market that maybe in the next years are going to be used for this purpose. But at the moment, these are the platforms that are used. Once the libraries are sequenced, the generated sequence require a specific analysis that Jorge will explain now.
I'm Jorge Amigo. I'm in charge of the data analysis of the information, and I'm going to share with you a couple of tools we developed. The first one is the first one regards the designing of the sequences needed to tag an entire library. When we started working with Dell libraries, we started working with a single set of tags that was already published. There wasn't everything else available, so we ended up building up our own tool to generate the sequences needed to tag all these compounds on each library. We ended up building this tool that was able to, once decided the sequencing length, we were able to force the sequences to be 1, 2, or even 3 different apart, which helps the sequencing process later. We could control the mass of the sequences by controlling the GC content. We can even avoid having reverse complementary sequences on the set, so that nothing is mixed when dealing with DNA. We can also avoid to have homopolymers in the sequences, and we can even avoid having custom patterns like restriction enzyme sites that we wouldn't like to have them on our sequences. As I told you, when we started working with DNA encoded libraries, there was only one set of tags published by Galaxo, and there were only around 1,500 sequences of 7 base long. They had reverse complementary sequences on them, and most of them, there were only one difference apart. So a sequencing error on that difference could get a compound being misled. We tried developing our own set of tags with 7 bases long, and we ended up with more sequences than the ones that Glasso had. We had that set of tags without any reverse complementary sequences on them, and we even had those sequences with at least 2 differences apart. So we could have a sequencing error, and we wouldn't be misleading any compound on the detection. This is regarding the designing. When we helped to design the experiment, and of course we also focused in the detection of the tags. In this detection of the tags, when we started working with DNA encoded libraries, there was nothing published. Every paper was referring to in-house developments, and we started working with pattern matching and with sequencing alignments, because it was what we knew how to do. But we ended up developing our own tool that it was to date the fastest and most accurate algorithm available. It is able to work in seconds or even just a few minutes. It scales linearly with the number of tags. That is very important, because usually if you work with big references, usually they scale exponentially, and it is quite an issue. And it also is very important that this method does not add any kind of error rate. So any errors that could be inside the data, would be due to the sequencing errors or even to the chemical errors when building the library. So the detection process does not include any error rate. So you will see, this tool is able to see exactly what is going down there. It is also sequencing technology independent. We have validated this tool with Illumina data and with ION data. And it not also describes what you expect to find, but it also describes things that you wouldn't be able to know if you were looking only by the things that you are expecting. This is very important when you are designing a library and when you are setting up an experiment. Regarding the expected results, of course, we are able to provide all the counts of the individual compounds. This, if you represent them in a 3D space, are all the points on that 3D space. But once represented the data, you can also see, as in the image shown there, lines and planes. These would be families of compounds. A plane would be a family of compounds that they share the same sequence tag, and a line would be a family of compounds that share 2 tags. So you can even select them by visual inspection. But also, we found out that it was very important for us to describe also things that were not expected, like repeated tags, unexpectedly repeated tags, that we call chimeras, or PCR duplicates that we are able to distinguish and to remove them by using the generated bases on the closing primers. And we can also detect sequencing errors. We can avoid them, or we can deal with them and study them. We can also look to reads that would be left out just by quality or by the length. But studying those left out reads can also help to develop a library, to set up a design. And once everything is perfectly described, you can start working just everything being done in the same way. Because the first experiment, sometimes you have to test how well your library develops and how well it performs, and then everything that is perfectly controlled, you can set it up just with high throughput. Okay, and now I'm finished. I think I have to share. Edu is going to share the last slides.
These are just a few questions that we recognize that are important for the field, and probably any of you familiar with the technology has tackled similar challenges with this technology of other. And also I can see that in the questions there are many other in the questions that from the attendees, those are also covered. And for us, it's very important if we can integrate the platforms, normalize the data, and develop the standards. Whatever you, the more integrated is the team, the better chances of success you have. Automation is also very important, not only for library production, but for affinity selection and sequencing. All this kind of analysis of the data drawbacks might have to do with correlating the DNA counts at the end that you get at the end with the potency of these molecules, which I think is still something that the technology cannot do. Also, when producing or generating a library, you want to make sure that the synthetic reactions provide sufficient yield. They are DNA compatible and are performed and some quality control steps by analyzing the content of your library by LCMS, for example, are done. And also there is a challenge with signal to noise ratio when selecting active compounds for resynthesis, because you are going to be resynthesizing a large number of different compounds, and you are going to, and this is going to be time consuming. So we think that integrated platforms, methods, and coordinating teams are needed. Of course, we still don't know whether these hits that are identified by this mean are going to impact in the long term the clinical pipeline. We know that there are many examples reaching clinical trials, but so far no compound identified by this technology has reached the market, which means that besides the technical challenges of the intrinsic challenges of the technology, we have to think about the drug discovery process entirely. Many of these challenges might be associated to the process. But in any case, there are compounds that have reached, and in terms of the expanding the drugability of targets or the ligandability, I think it's well proven that this technology can be applied to enzymes for the most part. The example of targets that successfully has been druggable with this technology, but of course, targeting DNA and RNA binding might be very challenging, as well as intracellular targets such as transcription factors and so on. I think it is demonstrated that the chemical space can be successfully expanded to unprecedented limits, making these libraries available for initial screens. And also, I think right now we are starting to see examples of peptides encoded by this technology.
Thank you, Eduardo, and thank you all for your engaging talks. So right after our last session, we are going to answer as many of your questions as possible. So Mabel, will you share with us a few examples of applications so we can have a practical overview on top of the theoretical background on the DNA encoded libraries?
Yes, thank you very much. We will be very happy to do so, right, Angel, to create this kind of collaboration for a chemical library on Dell in EU-OPENSCREEN. Very happy.
At this point, and before thanking you all for being here today, I would like to take this opportunity. Maybe I can share my screen now. It will be easier to show you where you can find us, where you can find EU-OPENSCREEN, and if you have any questions, just to visit our website or subscribing to our newsletter. So I will take this opportunity now to tell you that we would very like to hear back from you. So after the end of the webinar, a feedback questionnaire will be sent to you by email, will be sent to the attendees by email. So your feedback and comments are more than welcome in order for us to improve and advance our future training activities. If you would like to receive more information on EU-OPENSCREEN ERIC technologies and partner laboratories and services, please don't hesitate to sign up to our newsletter using the link indicated in the blue arrow, or just scan the QR code that you see here in our screen. If you want to know more about EU-OPENSCREEN, please visit our website, www.eu-openscreen.eu. So a very special and warm thank you to our expert speakers today. It was very, very nice having you here today for this interesting webinar. And a very warm thank you also to all of our attendees. We will hope that we will see you also soon to our next webinar and training activities. So have a nice day and hope you enjoyed our today's webinar. Thank you all.